Hello, my name is Alla Krasikova, and together with Veniamin Fishman and Tatiana Kulikova, we are presenting our review essay manuscript titled Lampbrush Chromosome Studies in the Post-genomic Era. Extraordinary extended Lampbrush chromosomes appear in the growing outsides of many animals except mammals. They are favorable objects in chromosome biology. Lampbrush chromosomes observed at the diplotent stages of oogenesis, so pairs of homologous chromosomes are joined at the sites of crossing over events, chiasmata, thus forming Lampbrush bivalents. The chromatin of Lampbrush chromosomes is packed into linearly arranged stable Hermannea loop complexes. Sister chromatids are condensed within chromomeres, while completely separated in the regions of lateral loops. Each chromomere loop complex may bear several lateral loops of different length, from microloops to extremely long loops, up to hundreds of micrometers. Lampbrush chromosomes are characterized by an unusually high rate of nascent RNA synthesis called hypertranscription. High transcriptional activity on Lampbrush chromosomes was demonstrated by different approaches, such as visualization of the nascent transcripts by incorporation of bromo-TP, retraction of lateral loops upon inhibition of transcription, and others. Lampbrush lateral loops represent the most prominent example of so-called transcription loops. The term transcription loops refers to looped out, transcriptionally active chromatin domains in various model objects, such as lateral loops in Lampbrush chromosomes, puffs in polyten chromosomes, Lampbrush-like loops in Drosophila spermatozoites, and highly transcribed genes in mammalian interface nuclei. One or several functioning transcription units with a polarized nascent RNP matrix can be clearly resolved in each lateral loop. It has been proposed that Lampbrush chromosome structure with extended transcription loops bounded by regions of compact chromatin can integrate into the general interface chromosome architecture. However, until recently, the information on the spectrum of sequences transcribed on the lateral loops of Lampbrush chromosomes was mainly limited to tandemly repetitive elements. The lack of genome-wide data on sequences transcribed on the lateral loops of Lampbrush chromosomes limited the possibility of discussing the biological importance of this phenomenon in animals with hypertranscriptional type of oogenesis. In our review, we highlight novel findings in Lampbrush chromosome studies in post-genomic era. To further advance our understanding of Lampbrush chromosome organization into functional domains, we consider a comprehensive study of the DNA methylation profile, nucleosome positioning, pattern of epigenetic modifications, and a distribution of chromatin contact domains using advanced whole genomic approaches, such as single-cell high-throughput chromosome conformation capture and bisulfite sequencing. These techniques will allow to create a genome-wide map of Lampbrush chromatin domains with different sets of epigenetic modifications. Moreover, new whole genomic approaches can be applied to study the regulation of gene expression at the level of transcription on Lampbrush chromosomes of various species. This new data can be combined by cytological visualization of different chromatin domains using state-of-the-art imaging techniques combined with immunofluorescence detection, fluorescent in situ hybridization, RNA fish with nascent transcripts, and fluorescent in situ sequencing with very high detail. We believe that this complex approach will help to close certain gaps in our understanding of the function of Lampbrush chromosomes in oogenesis. Unique opportunities offered by these spectacular chromosomes with the increasing technological advances will help to establish a link between the special organization of the genome and the regulation of transcription. To learn more, you can look through our manuscript. We would be delighted to know what you think about these directions. At the end, I would like to thank my co-authors, Veniamin Fishman and Tatiana Kulikova. We also would like to thank BioEssays for publishing our article and you for watching this video.